The electron transport chain, electron transport chain. So the electron transport chain again occurs in the mitochondria. And it is going to happen along the inner membrane and in the intermembrane space. Okay, this process is not going to occur within the matrix. So if I make a big cell here, I'm gonna blow this portion up. So here and here. So out here is the cytoplasm. You can orient yourself. This is the outer membrane of the mitochondria. This here is the intermembrane space. This is the inner membrane. And so down here is going to be the matrix. Now, we have a whole lot of electron carriers that we got from glycolysis and the Krebs cycle. Okay, and in fact, we ended up with 10 NADHs and 2 FADH2s. So what do we do with all these electrons? So remember, these are carriers, so they're holding on to high energy electrons. And we've got 12 pairs of them. Well, in the membrane, there are proteins. We know this, okay? So there are proteins there that are lodged in the membrane. And there's a sequence of them. And these proteins are little pumps. And when we use our electrons, we can use these electrons here, and we can, with these electrons, we can basically turn them into usable pumps. And what they do is when we use those electrons, they pump hydrogens from the matrix into the intermembrane space. Okay? Those electrons, once those proteins do that, they don't get completely used. They actually hop over to the next protein. And then that protein pumps hydrogens from the matrix into the intermembrane space. And then those electrons, with what's left of their energy, they jump down to the last protein And that protein does the same thing. It pumps hydrogens into the intermembrane space. So the intermembrane space is now filling up with hydrogens that are taken from the matrix. So now there's more hydrogens in the intermembrane space than in the matrix. So there's another little protein on here that is an enzyme. And it looks like a little machine thing here. And this is called ATP synthase. And in it is this crazy, weird little thing in here. It's like a little machine thing. And when the hydrogens come up here, and they can only go they're only pumped one way into the intermembrane space, we now have a gradient for hydrogen to go back into the matrix. So the only way for those hydrogens to get back into the matrix is to go through ATP synthase. So the hydrogens go through ATP synthase, and when they do that, it turns this little combine thing, this little machine, and when that happens, we get ADP plus inorganic phosphate attached to make ATP. This process where we're using this gradient of hydrogen is called the chemoosmotic process. Chemoosmotic process. I don't know if that's the right way to say it, but anyway, so that process couples the movement of a substance and in this case it's hydrogen, so the movement of a substance across a membrane to create or cause chemical reactions. Which in here, we're using hydrogen that's coming through this ATP synthase, and that's causing the reaction of ADP to inorganic phosphate to make ATP. And every single time a hydrogen comes down here, we get an ATP. That process is called oxidative phosphorylation. Phosphorylation. So this is oxidative phosphorylation that makes this ATP. So again, every time a hydrogen comes through there, we get an ATP. So in the electron transport chain, we end up with about 28 to 32 ATP, because each NADH makes about 2.5 ATP, and each FADH2 makes about 1.5 ATP. So that brings us to that 28 number, 28 to 30, whatever it is. Okay. All right, so looking at this picture, you may or may not notice this, but there's a problem here. And the problem is in these electrons, because what's been happening here, we took the first two electrons and we stuck them up here on this protein, and then they jumped over here, and as soon as they jumped over to the second protein, then we had the next pair of electrons coming to this protein. And then when that second pair jumped to the third protein, and then that next pair jumped to the second, and so on, once we got electrons on all three proteins, what happens? Can we still use the protons or the electrons that we have left? Not really, because there's nowhere for these people to go. There's nowhere for these electrons to go. They're kind of stuck here. And if they don't get off this protein so that this electron can come down and this one can come down, there's like a log jam. It just stops. So there has to be somewhere for these electrons to go. So we remember that the oxidation of glucose, we get the glucose is oxidized to carbon dioxide, but the oxygen was reduced to water. So we have oxygen down here. So those electrons now come down onto the oxygen, and it will make water. So that's where the water comes from in that equation. The oxygen is reduced by taking those electrons from that protein. So oxygen is called the final electron acceptor, because the electrons were already accepted by the electron carriers. They were already taken up by the carriers, and now they're being used on these proteins, and somebody else has got to get them. So that is oxygen. Takes up those electrons so that this electron transport chain can continue and we can finish making all this ATP. So in the end, then we end up with about 34 to 36 ATP. Per molecule of glucose. All right, so that is the end of cellular respiration as far as the oxidation of glucose. Now we'll look at how fats come into play.